my proposal for iRT, which is concerning. This is essentially a drug release study. So we're going to be trying to release lysozyme, which is encapsulated in PEO from a scaffold. So let me explain what exactly this is. So essentially what happens is you have a lysozyme, which is a model protein for a growth factor. Obviously, using tons of growth factors in experiments would be expensive and not necessary, so we're just going to use a lysozyme. That lysozyme is mixed in PEO, and the PEO protein, which is polyethylene oxide, is essentially encapsulating the lysozyme in essentially a form of like a microsphere, like a spherical shape. And so what happens is you have a polymer solution, so PVDF TRFE, which is polyvinylidene difluoride and trifluoroethylene, which is a plastic dissolved in a synthetic solvent called MEK, which is methyl ketone. And you put the lysozyme, which is embedded in the PEO, in the solution, so you essentially have growth factors embedded in a PEO protein, which is mixed in all this plastic. Essentially, what happens is you have all that solution in the syringe, and the syringe essentially, there's a syringe pump attached to the end of this, and we wouldn't be doing, I'm just trying to explain some background, we wouldn't be doing this at Hungary, but essentially the syringe pump is really releasing the solution at approximately 3 ml per hour. So there's like a tiny bit of fluid coming out at a very, very slow rate.
So you're generating basically this fiber that has lysozyme encapsulated in it.
Nanofibers with PEO embedded in it,
right.
Which has lysozyme inside of it.
Correct. Yes. Okay, got it.
But in a fiber rather than a
yes,
sphere?
A nanofiber. Essentially, so what happens is when you apply electricity to the end of this polymer solution that's coming out, it forms fibers, and there's a rotating collector over here which collects aligned fibers, essentially. So that's the background. Are there any questions on the background before I move forward?
I don't know why we're doing this, but okay. I'm assuming you're going to tell us why?
So essentially what the purpose of the scaffold is for neuronal repair. So essentially what would happen is you would put the scaffold, the neurons, or for example, like, you would put neurons on the scaffold, and basically, essentially what the scaffold provides for is physical guidance for the neuronal growth.
Now would the lysozyme be able to, if it were in fact...
So the lysozyme itself is a modeling
right,
the growth
right.
factor.
So
the growth factor, we're trying to get the growth factor out of the fiber. So basically, what my project would be concerning would be trying to get that the PEO protein out of the scaffold at a certain a prolonged rate, because essentially what happens is basically the optimal neuronal growth happens when it's prolonged, when there's a prolonged release for about 2 to 3 weeks. So what happens is you would want that, when you put the scaffold in the, and neurons on top in like an ideal condition in the body, PBS, which has all the ions, which are essentially, in a sort of a environment like similar to the spine, what would happen is, you would have that physical guidance with those nanofibers, the interfiber spacing, the fiber diameter, and the 3D structure essentially the scaffold is providing. A way for those neurons to grow. Secondly, this material is piezoelectric, which means that essentially with the natural deformation in the spine, essentially when you put the scaffold actually releases some surface charge, which essentially helps the neuron to grow. And thirdly, this basically trying to embed this lysozyme, which is essentially the growth factor, modeling the growth factor inside the PEO, which is inside the scaffold. If we can get that PEO out and the growth factor gets released, then it can essentially help the neuron.
So what would you be doing here?
So here,
before we get to that, just to make sure I'm clear, the purpose of all of this is to basically generate a new, this is a different way of providing a very specific organization for the scaffolding so that you can grow neurons in a very defined manner.
Yes.
Right, okay.
So we're going to be dealing with the release of the growth factor part of it in my project here at Pingree. So essentially what's going to happen is just kind of getting to the experiment would be, so I would have these scaffolds sort of already produced at NGIT where I did research.
So the fiber you'd get from somewhere?
Yeah, these scaffolds, the entire thing with the PEO, the lysozyme, everything inside of it. It's like essentially like a piece of cloth. You can think of it, but it's much more complex at the sort of the.
Got it.
But so you would basically cut circles of that scaffold, and you would wash it and sterilize it, and you would put it into well plates. So you can think of it. You would put it into, for example, a 96 well plate.
And then we're just measuring. So at these time points? What are we measuring specifically?
So
the lysozyme concentration in the solution.
Yes. Hold on, you're ahead of me.
Sorry.
We're putting the scaffold into the wells.
Mm-hmm.
Then the whole goal is to measure the PEO, whatever you say.
PEO, right?
PEO that comes out of the scaffold.
So essentially, we want to see how much growth factor comes out. So the reason why we're putting PEO lysozyme inside of PEO is because the growth factor would simply denature when it was mixed with that MEK solvent over here, because it's a synthetic solvent. It would just and also just to note when this is spun, as soon as it sort of like collects while it's being transferred from the electricity and it's being collected to the rotating collector, which is grounded, which is why biophysics-wise it's going there.
Mm-hmm.
The solvent evaporates essentially. Okay. So you need the solvent to make it in the first place, so you need to make sure that your growth factor is denature. So essentially, what we're trying to do is so we put PBS solution into each well, all these 96 wells, and so as Mr. D kind of mentioned before, so these time points are essentially, these are 11 time points, 1, 2, 3, 4, and 8, and 11. And so if you think of it as a sort of a 12 by 8, then you have 11 of them as different time points, just think of them for now, and you have one control group, sort of, just to see what's going on. So essentially what you would do is you would incubate them at 37 degrees Celsius, which is about 98.6 Fahrenheit body temperature, because you're trying to regenerate spine, so this is like the ideal condition you would want to test all this out in.
Mm-hmm.
And so basically what happens is at these different time points, you're gonna take the whatever is released in this PBS solution, so whatever lysozyme is released into the PBS solution, and you're gonna run, which I'll get into in a little bit. You're gonna basically run a sort of a test to see how much drug has been released, so how much lysozyme has been released.
How do you measure that?
So you use something called a lysozyme kit assay. So another reason why it's sort of convenient to use lysozyme over growth factors is because you can use this assay to sort of figure out how much lysozyme has been released. So at each of these time points, in each sort of each column, and sort of why we have 8 rows is just to do it over again. Take the average of them, so that. But essentially, what you would do is you would prepare the lysozyme standard curve. So you would basically see how much is there, and then you would essentially once you get down to the main part, you would essentially see how. So you would see how much you would basically try to measure lysozyme activity, which would tell you.
So you're measuring activity, not just presence of protein, but activity.
So you're measuring. So what happens is you put a substrate, right? You put a substrate which is conjugated with a bacteria, okay? In the solution. So you suck out the solution and
add bacteria to the solution.
So you're basically, the lysozyme is acting on the thing that the bacteria produces, and when it acts, it turns yellow.
Okay, so
this is a separate test. You pipette everything out, put it in, and this is just for the test, not for the scaffold. This is just to see how much lysozyme has been released. Okay. So
you take some out of your 96 well plate with the scaffolds, you take some solution out, whatever much, and put it into a new 96 well plate, basically.
Yeah.
That's your colorimetric assay.
Yeah. Actually, not all night. Yeah, you could put it in.
It's fluorescent. It's fluorescent.
Yeah. So what happens is when the lysozyme acts on the substrate and sort of the bacteria as well, what happens is it gives off light. So, and with that light, you can essentially tell what the concentration of lysozyme is, and then you can tell from there how much lysozyme exactly has been released. Is that?
Okay.
I'm good.
Why can't you just measure the amount of protein that there is?
Well,
I mean, active lysozyme would be nice. That's fine, but like, if the lysozyme is being secreted into the solution, couldn't you just measure protein concentration?
You could
Because that's easy.
But
easier. I'm just wondering why you would want to test.
But you're looking for active.
Yeah. Well,
activity.
no, not just. I'm literally looking for how much growth factor has been released, so that's why I want to sort of focus.
Right, which is why I'm wondering if it's like something in the solution that would prevent you from actually measuring protein concentration.
They've got to suspect that something in.
Yeah, I'm sure it has to be like. I'm just confused as to why, because it seems so easy to measuring protein concentration is no problem.
Yeah.
But.
I can maybe get back to you on that.
Okay. Yeah, I mean, I'd be interested to know because obviously that would make your life a lot easier.
Because I do know
having
that
a spectrometer is easier to deal with than a spectrophotometer.
Do you understand the question she's asking you?
She's basically asking me why am I doing this when I could be doing something easier.
She's asking you basically, do you suspect that something is going to happen to the lysozyme as it leaves the fiber?
Wait, is there something in the solution that you're taking out to add to this assay that would prevent you from being able to just simply measure amount of protein? Because you could use like a Bradford assay for measuring amount of protein, or even just absorbance reading.
Well, to my knowledge, I'm fairly sure that both the PEO and the lysozyme is released. So
maybe PEO influences in the visible light spectrum.
I mean, it makes sense though. What they're trying to.
You would want to essentially. So I think to answer your question, I think what would be the appropriate explanation for this would be that essentially when the growth factor, you want to make sure that the growth factor is also fully out of the PEO, which is what's suspected, right? And so you want to see
you want to see active.
Is the sort of the growth factor of the lysozyme able to act with the substrate?
Okay.
Not just like embedded in the protein, just comes out. So that's sort of
okay.
Cool.
Okay.
I'm okay with that.
And then so we'll move on to some of the costs.
Actually, I'm not okay with that. Can we go back? I have one more question.
Sure.
So you're detecting fluorescence. Where does the fluorescein come in the kit? What's actually fluorescent? The molecule that the lysozyme acts on?
Yes. Yes. And so that when they react, it essentially gives off light.
So you're going to need a spectrophlorimeter.
Yeah.
Fluorimeter for this.
Which I would need a spectrophlorimeter.
Okay.
And
they're like uber expensive.
Don't we already have?
The old specs should be able to handle this.
Not for fluorescence. We can't use a fluorimeter. The fluorimeter is a separate. When you're measuring fluorescence, it's not the same as a spectrophotometer.
Right, but so I was under the impression that when you were doing like luciferase or GFP, you would need one of those. But I thought with fluorescein, because of the
if it actually turns, you can see
that's
yellow,
what I'm saying. I think the signal was so strong.
You're measuring just color change.
Well, you're measuring the amount of a certain wavelength that's given off.
So I think what we just need you to answer later, and you're probably gonna have to look this up, is what exactly is the machine that will detect the fluorescein?
Well, that would be the spectrophlorometer, and
because I've used fluorimeters plenty of times for fluorescent molecules.
So she's asking you to distinguish between whether it would be a spectrophlorometer or a spectrophotometer.
Okay.
They just measure fluorescence versus visible light.
And if it's a spectrophlorimeter, does your collaborator have one that you could use?
Right.
I mean, they definitely do.
Okay.
But
where are they located?
NJIT's in Newark.
Where?
Newark.
Newark.
So
you could conceivably go over there and use their machine.
Yeah.
Right?
Yeah.
Possibly, yes. But I'll get back to you on that. And the cost. So just to kind of like clear. So this cost is obviously the lysozyme assay kit. Each kit performs about 400 assays. For about if we were to do one drug release study, which would consist of a 12 by 8, which would be 96, times 3 different samples just to sort of verify. So 3 different scaffolds. You would basically use 3 different mats altogether because there could be some variability in the electrospinning process, perhaps. And then doing each of those twice, so the whole thing twice, at the same time, of course, would lead to about 538 assays, which is about 1.33.
So we'd be able to run 2 full experiments.
With this kit? Okay.
What? So I would only be able to run, I would need about 4 kits to run 3 experiments.
4 kits to run 3 experiments. $4,000.
This is the cost of 4 of them.
Ah, thank you.
Each one is about
total cost
$1,000.
Oh, yeah, you can have that.
Each one is about
You
$250.
can take pictures of this.
So, I'm assuming the lysozyme, the reason you're using lysozyme is because that's what they're using. This is how they're doing it. Right? I mean, you didn't propose lysozyme, this comes from them.
Yes.
Okay. Because there's so many other enzymes I could think of that are easy to work with.
They've got to be doing it. There's some reason they're doing it.
Yeah, there's got to be a reason.
Well, lysozyme is also cheaper. That's also one reason. Right?
Cheaper than what?
Well,
She's not talking about the lysozyme. She's talking about the fluorescein.
I'm talking about the kit, and the measurements. That's the kit's kind of expensive.
okay.
And the measurements, why you would use fluorescence if you could just do something simpler.
If I recall correctly, the measuring level of PEO was actually more expensive or more difficult.
Okay, that's fine, but it's the enzyme that I'm wondering. Because, for example, in the research class we use lactate dehydrogenase. The kit to measure LDH is $100, and I can use it for a lot of reactions, right? So that's all I'm wondering.
It might be just that lysozyme is functional groups which are similar to the growth factors BDNF and NT3 that we're working with when you're on growth factors.
So then my other question would be I'm assuming they're doing these experiments too?
Yeah.
And this is just more data? Or is there something different that you're doing that they're not doing?
So it would be more data, but so hypothetically what would happen is if you use the experiments from the results from this experiment, some of the things which are modified are in the electrospinning process. There are about 5 to 6 different parameters you can modify there. So you're trying to see which scaffold type will lead to the maximum growth.
Are you asking whether it's novel?
We might also change the protein hypothetically if for some reason the drug release is not where we want it to be. Maybe looking into another protein might be something. So.
Well, I guess my question is really just: are they the people in whoever you're collaborating with? Is this just a side project for them, and they're just not really working on it, or are they actively doing these experiments in their lab?
They are actively doing these experiments in their lab.
So, what are you doing that's different than what they're doing?
So essentially, they would be doing. They are performing these assays, but essentially, if depending on the results of this, how the drug release study goes, I could propose perhaps, or go to NGIT and electrospin mats with different protein, for example, or a different sort of setup, and then run this.
So there could be some novel parameters. Yeah. There could be some novel aspects of it.
So this would just be more like a control, set it up to make sure in your hands you could get it done here instead of there. It just seems like kind of a lot of money to repeat the experiment that they're already doing.
Yeah.
That's my only concern, I think. Because I think the experiment makes sense, and I definitely think that this is.
Yeah, it's a well presented, well thought out, well.
I'd like to see you be doing something different from what they're doing. So whether it was a different protein or.
Yeah.
Wait,
so
I'm getting really tired.